So what you can see on this robot platform are stacks of microtiter dishes. They're essentially plastic plates that have lots of little wells in them. One of those stacks of plastic plates contains yeast cells which harbor a plant transcription factor. These transcription factors are the on-off switches of the plant genome. They're going to be very important for us to understand basic biology of how plants grow and adapt to environmental conditions, and they're going to have practical applications of how we can confer traits onto plants that are going to be useful agriculturally. So what our robot is doing here simply is mixing tens of thousands at a time different yeast strains, and it's asking actually a very simple question. Which genes do these individual transcription factors, all 2,000 of them, bind to and regulate? The robot can do overnight what it would take tens and tens of graduate students to do in several weeks. So we take advantage of the robot's ability to manipulate and move these liquids around at high speed in a high throughput format. That enables us to do tens of thousands of experiments that would be beyond the reach of an individual grad student. And so the grad student now can focus on the data that the robot generates rather than moving thousands of liquids around for several weeks. So what we expect to learn from this is a basic wiring diagram of how plants grow and develop, of how plants survive in different environmental conditions. And we think ultimately we're going to really learn which of these transcription factors are going to be interesting for developing new kinds of crops. Crops that are going to be resistant to climate change, crops that are going to be more nutritious, crops that are going to grow under marginal conditions that we see in areas of the developing world. So we've generated a unique resource. We are going to make this resource freely available to researchers all over the world, and we expect this to have significant impacts in understanding the basic functions of plants and translating that information into agriculture.